Deduve and co-workers isolated in 1965 particles from liver cells and other tissues which were enriched with some oxidative enzymes, such as peroxidase, catalase, glycolic acid oxidase and some other enzymes. The name peroxisome was applied because this organelle is specifically involved in the formation and decomposition of hydrogen peroxide in the cell. These are single-membrane enclosed cytoplasmic organelles found both in animal and plant cells. These are characterized by containing H2O2, producing oxidases and catalase. They are approximately 0.5 nanometers in diameter. They have also been found in protozoa, yeast and many cell types of higher plants. Peroxisomes play a crucial role in various metabolic processes, primarily by carrying out oxidative reactions. Oxidative reactions and detoxification. Peroxisomes are specialized in performing oxidative reactions using molecular oxygen. A significant byproduct of oxidative reactions is hydrogen peroxide, which is highly toxic to the cell. To neutralize this toxicity, peroxisomes contain a high concentration of the enzyme catalase. Catalase rapidly breaks down hydrogen peroxide into harmless water and oxygen. Fatty acid metabolism, beta oxidation. Peroxisomes are major sites for the breakdown of very long-chain fatty acids and branched-chain fatty acids through a process called beta oxidation. While mitochondria also perform beta oxidation, peroxisomes handle the initial breakdown, converting them into shorter-chain fatty acids that can then be further processed in mitochondria for energy production. This is a significant source of metabolic energy. Lipid biosynthesis. Peroxisomes are involved in the biosynthesis of certain lipids, including cholesterol and dolichol in animal cells. They play a critical role in the synthesis of plasmalogens, which are a special class of phospholipids with ether linkages. Plasmalogens are particularly abundant and essential components of myelin in nerve cells and also found in heart tissue. Role in plants. In plants, specialized peroxisomes called glyoxysomes are found in germinating seeds. These convert stored fatty acids into carbohydrates, which is crucial for providing energy and raw materials for the growing seedling before it can perform photosynthesis. Peroxisomes in plant leaves are also involved in photorespiration, a process that metabolizes a side product formed during photosynthesis.